You may know about pathway. A pathway corresponds to a series of action that can lead to a change in the cell or a certain product. That is, it can produce protein or can turn on or off a gene. So what we will talk about, how can we extract the available pathway in the KEGG database. So we only focus on the human pathway. So to extract the pathway from KEGG database, we need a few library. That is, we need the library Lima, then the database. This database is for Homo sapiens, and the annotation DBI library. So all the library you can install from the BioInnoTech manager. So let's load this library. It's loading. So after loading, we will extract the gene KEGG links. So it will faces the pathway ID and the corresponding interest ID. It's running. So after finishing, we can see what is inside the gene KEGG links. So it's finished. It has total 37k plus objects. So here are two things, that is the interest ID, interest gene ID, and the pathway ID. Now we will link up this interest gene ID with the gene symbol. So to do this, okay, if we look at here again, we have another column that names symbol, and it contains the corresponding gene symbol name of this interest gene ID. After, we will extract the pathway names. So we have total 359 pathway available in the KEGG database for Homo sapiens. These are all pathway. So we can see the name: metabolic pathways Homo sapiens human, carbon metabolism Homo sapiens human, and there are many pathways available. Now what we will do, we will merge these pathway names with the gene KEGG links, so we can write them, merge them in a same database, in a same data frame. Okay, so if we execute this, we have the merge df, and if we want to see the first few columns, so we have there we can get pathway ID, then our gene ID, then the gene symbol, and description means the pathway name. And now we can save this result for our future use. So this is the name, human KEGG pathways genes.csv. This is the final output file name. So you can look at the result in there also. And if we want to look at the data frame here, so here we can see this. So this is all for today. Thank you for watching.